Today in the news, we'll be hearing about the EU approval of a second-line regimen in non-small cell lung cancer, the extended FDA review for multiple myeloma treatment, and results from a prostate cancer trial. In addition, we'll hear about how the presence of PD-L1 may predict response to treatment and a potential genetic basis for tamoxifen resistance. All that and more starts right now on OncLive News Network. Hello and welcome. I'm Laura Jones.
And I'm Justin Gallagher.
The EU has a new second-line treatment option for lung cancer patients with advanced adenocarcinoma after first-line chemotherapy. Nintedanib was approved at the end of November 2014 to be used in combination with docetaxel. Approval was based on results from the LUM-Lung-1 trial, which enrolled 1,314 patients with non-small cell lung cancer who had already received first-line chemotherapy. Results published in Lancet Oncology in early 2014 demonstrated for patients with adenocarcinoma, nintedanib, when added to docetaxel, significantly extended median progression-free survival at 3.4 months versus 2.7 months, and median overall survival 12.6 months versus 10.3 months when compared to docetaxel alone. Additionally, approximately a quarter of patients receiving the combination survived for 2 years or more. In terms of safety, nintedanib demonstrated a manageable adverse event profile without further compromising patients' overall health-related quality of life. According to Dr. Martin Reck, lead investigator of the LUM-Lung-1 trial, the approval of nintedanib offers a much-needed new treatment option for adult lung cancer patients with advanced adenocarcinoma in a second-line setting.
Turning to the United States, the FDA has extended its review period for panabenostat in combination with bortezomib and dexamethasone for patients with previously treated multiple myeloma. This extension follows a 5-2 vote against the accelerated approval of the novel HDAC inhibitor by the ODAC in early November of 2014. The committee's decision was based on the mixed results from the Panorama-1 study, in which 768 patients were randomized to bortezomib and dexamethasone with either panabenostat or placebo. Panabenostat-treated patients had a significantly longer median progression-free survival of 12 months versus 8.1 months with placebo. However, secondary measures, including overall survival and overall response rate, were similar between the two arms. Addition of panabenostat was associated with increased frequency of adverse events, the most common being grade 3 and 4 thrombocytopenia, reported in 67.4% with panabenostat versus 31.4% with placebo. According to Dr. Richard Pazder, Director of the Office of Hematology and Oncology Products within CDR, progression-free survival was not the question. There's clearly a benefit. The question is, what is the magnitude of benefit, and does it warrant the toxicity? A final decision is expected in early 2015.
There's disappointing news for patients with prostate cancer. The COMET-2 trial of cabozantinib being tested for bone pain in men with metastatic castration-resistant prostate cancer did not meet its primary endpoint of alleviation of bone pain. This double-blind, controlled trial randomized 119 patients to either cabozantinib or mitoxantrone and prednisone. 15% of patients in the cabozantinib arm reported a pain response compared to 17% of patients in the control arm. Following the disappointing results from the COMET-1 study, Exelixis halted its enrollment process for the COMET-2 study. Randomized phase 3 trials, pivotal trials of the cabozantinib continued in metastatic renal cell cancer and in advanced hepatocellular cancer.
The presence of an immune-suppressing protein in noncancerous immune cells may predict how patients with different types of cancer respond to treatment. Dr. Roy Herbst, Ensign Professor of Medicine at Yale Cancer Center, led the multi-center phase 1 trial to test the efficacy of MPDL-3280A, a drug that blocks PD-L1, a protein expressed by many tumor types. The trial included patients with melanoma or cancers of the lung, kidney, colon, GI tract, or head and neck, whose tumors were evaluated for PD-L1 expression. According to Dr. Herbst, we knew that the expression of PD-L1 in tumor cells is critical in blocking the immune system, so we were intrigued to find that the expression of PD-L1 in non-tumor cells such as macrophages, also predicted drug response. Of the 175 patients enrolled, 21% showed partial or complete response to MPDL3280A. Across all tumor types, in patients with high PD-L1 expression on non-tumor cells, 46% showed a partial or complete response. Also notable was the finding that smokers responded better to the drug than non-smokers. This result could provide further insight into how smokers and non-smokers respond differently to drugs.
On the opposite end of the spectrum, resistance can play a major role in treatment choices. Johns Hopkins researchers believe they may have found a genetic basis for tamoxifen-resistant breast cancer. Mutations in the macro-D2 gene appear to provide one possible explanation for this resistance to tamoxifen. In one study, the scientists used RNA molecules to silence the gene's expression. What they found is that blocking macro-D2 restored partial sensitivity to tamoxifen, suggesting a possible new therapeutic target. In the analysis, macro-D2 overexpression was present in the majority of metastases in patients with tamoxifen-resistant tumors. Additionally, patients who had macro-D2 overexpression in primary breast tumor had significantly worse survival than those who did not. The researchers cautioned that there may be other genetic factors that control or contribute to tamoxifen resistance, and that nothing in this study should suggest that tamoxifen should be avoided. And that is the oncology news for this week. I'm Laura Jones.
And I'm Justin Gallagher.
Thanks so much for watching OncLive News Network. We'll see you next time.